So hello everybody, I think that was the starting sign-up. So thank you all for coming. I'm Felipe. I'm the first year PhD student in the Radcliffe Lab. And today I'm going to be talking about the evolution of synchronized multicellular development during long-term evolution experiment. So cell synchronous division during early development is a hallmark of of multicellular development. So here we can see that we can see it shown in the starfish embryo, the salamander, and even in the fruit fly embryo, and also even in green algae. And cell synchronous division is thought that is important because it helps improve the conciseness and robustness of the timing of events during early development, like the prezygotic genome activation. And in this case, like it's not known how cell synchronous division evolved. So I'm going to try to explore that question, but because this is not like a comparative evolution, so a comparative biology session, like you know that I'm going to be doing experimental evolution. And in this case, we are going to be using, well, in the lab we work with the model organism of the snowflake yeast or the multicellular yeast, which has been evolving for thousands of generations using experimental evolution. And something remarkable about this organism is that it started developing with non-synchronous division. And once synchronous division evolved, it has been maintained for thousands of generations. So this organism is going to allow me to study how synchronous cell divisions evolve. So first of all, let me back up because we all know that yeast is actually unicellular. So in the lab, we work with Baker's yeast, and in the lab it was achieved to make the multicellular phenotype was by doing a daily transfer in which there were two selective pressures. So the first one was a selection for faster growth rate with the 10% dilution, by performing a 10% dilution. And the second one was by doing a settling selection in which they were selected for a larger size, and only the bottom of the tube was transferred to the next growth cycle. And if you do this process for several weeks, you will get this multicellular phenotype that we call the snowflake yeast, and this is caused by the ACE2 mutation that prevents cell separation during cell division. And this single mutation also gives the organism a multicellular life cycle where the cells are going to continue to grow, and the internal forces are going to start accumulating, which are going to cause the internal stress to accumulate, and one of the cell-to-cell connections is going to fracture, and that is going to give you the group reproduction, and just by continuing this, you get the life cycle. So this project started when I was doing an internship in the lab, and I was doing these time lapses of, to try to measure the doubling time of the cells. And how I was measuring the doubling time was just here by looking at the first frame where the bud appears and just counting until the next bud appeared. And in this case, we were surprised at how synchronous the divisions were in the evolved time points. And how I'm looking at the synchronous divisions is by comparing the doubling time of the mother and the daughter cells. And a way of visualizing this data is by plotting what is the mother and the doubling time in different axes. And here, if the cells are dividing synchronously, they are going to fall in the diagonal. If the cells are dividing, if the daughter cell is dividing more slowly, it's going to fall on top of the diagonal. And if the mother is dividing more slowly, then they are going to fall below the diagonal. So we can see that for the ancestor, where actually you cannot see it, but there are some points here below the diagonal. So the daughter cells are dividing more slowly than the mother cell. And probably these points you can actually see them, at least some of them, that for the evolved lines as early as 200 days of evolution, all of them are dividing synchronously and just falling in the diagonal, just with a little bit of variation. So, and a different way of visualizing this data, that this one you can see it, is that for the ancestor, if we just look at the doubling time distributions by the division number, or how many times a cell has divided, we can see that for the ancestor, the first division is taking longer than the rest of the divisions. So this delay between the first and the second division is what is causing what you cannot see in this plot, that the daughter cells are dividing more slowly. And this is something that as early as the 200 days of evolution gets rid of, that there's no delay of this first division. So Uh, to explore how cell synchronous division evolved in the snowflake yeast, we have two hypotheses, each one for each of the selective pressures that there are in the daily transfers of the organism. So for the selection for growth rate, cell synchronous division could have evolved as a cell-level adaptation. Synchronous cell division could be a side effect of selection for faster growth rate. And the other hypothesis that it could have evolved as a group-level adaptation, as synchronous cell division could result in a more efficient space filling, which could allow to increase the group size and also the fitness of the group. So in order to explore this, in order to test this hypothesis, what I have been doing is doing a network-based approach. And the reason why I'm doing the modeling is because the snowflake yeast already grows in a tree network pattern. So it's easy to approach them by using networks. And also because this will allow me to isolate the effect of changing the doubling time distribution without having to do an in vivo experiment of changing the growth rate or the cell cycle that can have confounding effects with other, confounding effects. So this model is made to be as simple as possible. The cells are represented as nodes, and the cell-to-cell connections are just represented as edges. And it also, here the cells can divide as indefinitely. There's no spatial restrictions. And the only parameter that the model is going to use is just the empirical doubling time distributions, where I'm just going to sample the next doubling time of the cell. And if a cell is dividing more than 2 times, then it's just going to sample from the last distribution. And the only part that I'm missing to be able to simulate the multicellular life cycle is just the fragmentation, which in this case I'm just going to be using what we term the H degree, that the H degree is just the sum of the node degree of the two nodes that form an edge. And a node degree is going to is just how many connections a node has. So in this case, for the node 2, it has 4 connections. So if I just add up the node degree of both nodes, and it goes above a threshold, I'm going to get rid of that edge, and that would be the group reproduction. So at first, this delay that the ancestral strain has is really bad adaptively, like it's not beneficial for the cells. So to explore what would be the benefits of getting rid of this delay, I have the doubling times of the ancestor, and I was also doing the other simulations with synthetic data where I got rid of the delay of the first division. And here we can see that if we just look at the growth rate, getting rid of the delay will give us an advantage. But as the title says, like I'm saying that the synchronous cell division evolved by a side effect of selection for faster growth rate. But you could say, like, maybe this is not something appropriate, like I'm just creating some synthetic data and this doesn't show a side effect. So really what shows the side effect is looking at one of the evolved strains. And here we can see that the evolved strain gets rid of the delay, like it also decreases the variation and gets, decreases the mean of the doubling time. And this one has a higher it has a higher growth rate. So this one will actually show that cell synchronous division could have evolved as a side effect for faster growth rate. But in the other hand, this is like the benefits on the cell level. At the group level, there could also be some benefits. So using these simulations, I was allowing them to grow and do several fragmentation events to keep track of their size at reproduction, because the bigger the cluster is, the more likely it is going to survive the settling selection. And here we can see that both of the synchronous dividing strains have a higher fracture size, a higher size at reproduction than the ancestor that is dividing non-synchronously. And this is like a little bit difficult to understand, like why is the case. So just like with the toy model, we can see that if all cells are dividing at the same time, they are going to be able to fill the space more evenly. And we can see that there are going to be some overlaps like happening at the same time with the cells labeled with the number 5. And if we compare it with a strain in which the first division takes twice as long, we are going to see that they are not using that, not filling the space as evenly, as there's going to be like only one point where they are running out of space. So, and also this is going to happen like at a smaller size. So really dividing synchronously will allow the clusters to reach a higher size, which will allow them to be more fit. So right now I have just showed that there are benefits at the cell level, at the group level, but I haven't answered like what is the selective benefit for each of them. So in order to answer that, I need to be able to simulate the whole daily transfers of the snowflake yeast. So I can already simulate the growth phase using the network simulations, but I'm missing to be able to simulate the settling phase. So in order to do this part, I'm going to use Stokes' law just to calculate the sedimentation rate of the snowflake yeast. And to be able to use this equation, I need to calculate the volume of the clusters. And for this, I'm just using 3D simulations to get their diameter. And with that, just to simulate the settling phase, I'm just going to sample at random a position where they are going to start, let them settle, and just stop the simulation when 10% of the final biomass reaches the bottom of the tube, and with that, start the next cycle. So with these simulations, I can do a competition essay to test what is the effect of the delay in both the cell level and the group level, and which one provides a higher selective benefit. And so I'm just starting both straits at a 50-50 ratio and performing several serial transfers. And in this plot, we can see that in the selective benefit or the selection rate, both of them are going to have a positive value, which will mean that the ancestor without delay is going to outcompete the ancestor in both the growth and the settling phase. So with this information, I can finally answer like which one is the one that is driving the evolution of cell synchronous divisions. And really the answer is not A or B, is that both can actually drive the evolution of this trait, given that both of them have a positive selective benefit. So in this case, I'm not actually answering which one exactly drove the evolution of this trait, but I'm showing that either one or both can be driving this evolution. And further work will be necessary to actually disentangle which one drove the evolution of the trait. So this is like really the main takeaway of my results. And like other takeaways are that, that synchronous cell division ensures the robustness in the timing of events, and it also improves the traits of the developing multicellular organisms, as this trait can allow the embryos to have a better coordination of the events without actually having to communicate the cells between each other. And also really, like something that is really important of this approach using experimental evolution and the snowflake yeast is that it can allow us to study fundamental multicellularity traits, like in this case, the cell synchronous division that would be really difficult to explore without this organism. So with this, I just want to acknowledge everybody in the lab for their help, including my supervisor, Will, and also Peter Collin, who has been mentoring me for the 2 years I have been in the lab, and Tom Belper for the feedback in the project, and also Tom Day for his help with the 3D model. So thank you so much. Are there any questions? Yeah.
Your simulations, you able to test them when used?
Well, I can tell you that the simulations, especially the fragmentation, won't approximate how yeast actually fragments. So like something that I'm developing with Tom Belper is like a more realistic model for the fragmentation, because it's not it's not easy to actually be able to figure out which edge is the one that in which fragmentation is happening. So yeah, like that's something that we are just trying to make it, and as I was saying, like simple as possible. And we hope that this model will still resemble some of the behavior that actually happens in the strain in the lab.
My name is Autumn, and I go to Georgia Tech. And today I'll be talking about disentangling the evolutionary impacts of relatedness in facultative versus obligate life cycles during the transition to multicellularity. The evolution of multicellularity was transformative for life on Earth. It evolved independently at least 50 50 distinct lineages, creating diverse multicellular organisms. Complex multicellularity evolved in 5 lineages: plants, animals, red algae, brown algae, and fungi. And they all share a trait in common in that they're all clonal, meaning that cells stay together after division to form groups. And so clonality is pretty well explained by social evolution theory in that the high genetic relatedness reduces the potential for within-group conflict. Now, these 5 lineages also have another trait in common, in that they're all obligately multicellular. And what this means is that their life cycle starts with a brief stint as a single cell, followed by growth and reproduction occurring in groups. Now, this is 1 out of 2 life cycles, the second being a facultative multicellular life cycle, where growth and reproduction occurs in cells, and growth and reproduction occurs in groups. And so they interchange between the two. Now, looking at these 2 life cycles may actually be impactful because the presence of a single cell stage may constrain multicellular adaptation. However, this is challenging to do because there's a lack of natural model systems that purely look at obligate versus facultative life cycles. And so to overcome this limitation, we used a combination of synthetic biology and directed evolution experiments to look at the role of obligate versus facultative life cycles using Saccharomyces cerevisiae. So our lab's main model system are snowflakes, and we currently have an ongoing evolution experiment that looks at snowflakes in this obligate life cycle for thousands of generations. Snowflakes can be formed via deletion of the ACE2 gene. And so for the obligate multicellular life cycle, you have your snowflakes. They reproduce by breaking off branches of cells, and they continue that pattern for the entirety of their life cycle. Now, in order to construct a strain that is capable of growing as single cells and as groups, we place that ACE2 gene in front of LEX-A, which is a transcriptional activator that turns on and off the ACE2 gene via the presence or absence of beta-estradiol. When beta-estradiol is added into the media, the ACE2 gene turns on and the population grows as single cells. When beta-estradiol is absent from the media, the ACE2 gene turns off and the population grows as snowflakes. And so for the facultative multicellular life cycle, you have your growth as single cells, and then some generation later, you have your growth as groups. And just a reminder that these snowflakes here, they grow via clonal development. Now, the single cell stage in the facultative multicellular life cycle may be more advantageous than the multicellular phase. And this is because there are more evolutionary units as a cell as opposed to groups. And so natural selection may be able to act more efficiently during the single cell stage in comparison to the multicellular stage. And so if we visualize this, let's say we have the same starting population. You have your single cells and you have your groups, where one group comprises of 20 cells. Well, the effective population size of groups are going to be 20-fold lower than the effective population size of cells. And so the number of genetically distinct lineages decreases by the size of the groups. So plants, animals, for instance, are going to have a much lower effective population size. Now, during this long-term evolution experiment, snowflakes evolved to form large, more tougher groups. And they do this through 2 key ways. One is the evolution of tetraploidy, which increases cell size. And the second is the evolution of cell elongation, which allows for entanglement of cells to form those tougher groups. Now, these are examples of multicellular traits that may come at a cost to growth rate and thus single cell fitness. However, if the single cell stage has a greater effective population size and natural selection may be able to act more efficaciously during the single cell stage, then the presence of a single cell stage may constrain multicellular adaptation. And so I looked at 4 different life cycles, one that is obligately unicellular, one that's obligately multicellular, and 2 facultative multicellular life cycles. one that spends 50% of its time as a uni, followed by 50% of its time as a multi, and that is called the U50-M50 life cycle, and one that spends 25% of its time as a uni, followed by 75% of its time as a multi, and that is the U25-M75 life cycle. During the unicellular phase, there is a daily selection for faster growth, And then during the multicellular phase, there is a daily selection for both faster growth and larger groups. So the first thing I wanted to do is see whether or not there is a difference in effective population size between single cells and groups. And what we found is across the life cycle, there is about a 10-fold lower effective population size in the groups in comparison to the single cells. In addition, the facultative multicellular life cycles follow the distribution of the stage that they are in. So for the first 4 days of the U50-M50 life cycle, it is a uni, and then you see it drop about 20-fold at day 5, where the remaining 4 days, it grows in a multicellular stage. And we see the same thing for the U25-M75 life cycle, where the first 2 days it grows as a uni, followed by the remaining 6 days, population grows as a multi. So not only do we see a difference in effective population size between cells and groups, but this also shows that the facultative multicellular strain works. And so now getting into the evolutionary outcomes of these different life cycles. The obligate multicellular life cycle has a much larger cluster size in comparison to the U25-M75 life cycle. And so spending only 2 days as a single cell is making a huge difference, not only in cluster size, but also cell size too. And so the obligate multicellular population has a much larger cell size in comparison to the U25-M75 life cycle. Now, ploidy may actually explain this. And so in the obligate unicellular population, it starts as a diploid and remains diploid over the course of 96 days. The obligate multicellular population starts diploid and evolves tetraploidy in as early as 56 days. The U50-M50 population remains diploid, but what about the U25-M75 population? Again, a population that spends only 2 days as a single cell. Well, that population also remains diploid. Only the obligate multicellular population evolves tetraploidy. And some characteristics of tetraploidy and Saccharomyces cerevisiae are 1, larger cluster size, and also large cell size too. Now, as I mentioned, tetraploidy is an example of a multicellular trait that may come at a cost, or they may lower growth rate and thus single cell fitness. And so here I wanted to directly test that. So I looked at relative fitness of tetraploidy in both a unicellular and a multicellular background. And here I used the selection rate constant, where R equals 0 means that there is no fitness benefit. In a unicellular background, tetraploidy has a small cost of 0.065 per day. In the multicellular background, tetraploidy gives a large benefit of 1.17 per day. Now, taking this a step further, I took the selection rate constants and created a simple model to estimate tetraploid frequency across the life cycle. So for the obligate multicellular life cycle, it's receiving that large benefit for all 8 days. For the U25-M75 life cycle, it's receiving that small cost for the first 2 days, followed by that large benefit for the remaining 6 days. And so I repeated this for the U50-M50 population and also the obligate unicellular population. And what I found is that only the obligate unicellular population sees a decrease in tetraploid frequency. Both facultative multicellular life cycles sees an increase, a huge increase in tetraploid frequency. And so despite this model suggesting that tetraploidy should be highly adaptive in the facultative multicellular life cycles, it doesn't happen. We don't see tetraploidy in the facultative multicellular life cycles. They remain diploid for over 96 days. So why don't we see tetraploidy in the facultative multicellular life cycles? Well, before I get into the hypothesis, I did 2 controls just to ensure that the system was working properly. So the first concern was that tetraploidy was evolving not because of multicellular adaptation, but as some feature of the obligate life cycle. And so Genesis, my mentee in the lab, she took the obligate multicellular strains, re-evolved them, and found that tetraploidy only evolves when you are selecting for larger size. The second issue was that there was a concern with the strain itself, in that once tetraploidy evolves, it impedes the alternation between the unicellular and multicellular phase. But however, that is not the case. Once beta estradiol is added into the media, the once tetraploid population begins to grow as single cells. So there's not an issue with the strain itself. So why we don't see tetraploidy in the facultative multicellular life cycles? We hypothesize that this is an invasion from rare problem. So let's say we have a starting colony where you have one tetraploid in green. Over time, that one tetraploid becomes 4 tetraploids, but it's still the same cluster, so it's formed clonally. Well, during our selection regime, only 1 out of 33 clusters are transferred each day, so there's already a low chance that tetraploidy, or this colony, in instance, is selected for. And then when it alternates into the unicellular phase, selection is actively acting against tetraploids. And so if you compare just the low chance in the multicellular phase and then being actively acting against it in the unicellular phase, it's really challenging to see tetraploidy to evolve and persist in these populations. And so I'm currently performing more tests to further test this hypothesis. So overall, complex multicellular organisms are both clonal, but they're also obligately multicellular. There's been a lot of work done looking at clonality, but little work has looked at the role of obligate versus facultative life cycles. And so we examined this by engineering a new model system and performed directed evolution experiments comparing the two. And what we found is that the presence of a single cell stage constrains multicellular adaptation with 2 key mechanisms. One, a single cell stage where selection is acting on single cell traits may conflict with multicellular traits. Is that the 1 minute or the 1 minute? Okay. Oh, is that the start? What? It's finished? Okay. Natural selection heavily favors the unicellular stage. And so with the snowflakes and groups, you have a much lower effective population size. So even highly beneficial traits are challenging to evolve. And then overall, obligate life cycles remove the potential for selection to act on cells. And this may explain why all lineages of complex multicellularity are also obligately multicellular. And then I'll just keep this quick. Thank you to everyone at Radcliffe Lab.
Hi, everyone. Thank you for coming to my talk. I'm a postdoc at University College London with Max Reuter and Ida. And I'm going to talk to you not about yeast experimental evolution, but Drosophila experimental evolution, and how we're trying to experimentally characterize adaptive conflicts over sex-specific phenotypes using our system. So sexual selection is quite a potent evolutionary force, shaping a great diversity of phenotypes in animals. And just the slide, it's quite beautiful and I like it. And it shows you the striking dimorphisms that you see in animals observed across the animal kingdom. So you have diversity not only in terms of body size, but you have complexity in anatomical structures, you have sex-specific ornamentation in some of these cases, behavior, shape, and so many different things. However, the evolution of sexual dimorphism is actually constrained by a shared genome. And what I mean by that is males and females, although they have these exorbitant different phenotypes, they essentially share the same genome. So what is happening? So sexes, as I said, have a high degree of overlap in their genome, and many traits are expressed both in males and females. And this generates the positive genetic correlation in traits, which I show in that figure there. And however, there are these fitness optima for several of these traits that are actually sex-specific. And what I mean by that is that traits which are that increase fitness when expressed in one sex may not do so when expressed in the other. right? And this can happen because some of these traits might be under stronger selection in males versus females. So in a sense, two sexes, despite having the same genome, they might have different optima for different traits, right? So this opposing selection pressure on males and females often results in maintenance of these genetic variants that are beneficial to one sex and not so much to the other. And this gives rise to sexual antagonism, which wherein different alleles at a given loci are favored in one of the other, or in two of the sexes. And this is what I'm showing you here. So there's this negative, the combination of genetic correlated traits, and the divergent selection leads to this pool, right, the constraint. Now, this phenomenon of sexual antagonism is actually quite ubiquitous across many species across the animal kingdom. You see it in fish, you see it in plants, and everywhere. And it is quite an important evolutionary force. Sorry, I'm trying to see if there is a pointer. Yeah, there is. Okay, sorry. So it is quite an evolutionary force, and the balancing selection generated by antagonistic fitness effects maintains a degree of maladaptation in which neither of the sex can attain its optimal phenotype. So it plays quite an important role in the maintenance of fitness variation, and it's an important driver in the evolution of sex chromosome differentiation as well. However, despite its fundamental role as an evolutionary driver and the prevalence of this across a lot of animals, we still don't know what is the genetic and a functional basis of sexual antagonism and what are the evolutionary dynamics. So it kind of still remains a bit poorly characterized. So two of the key questions that the field still lacks clarity in is, one, how many genetic variants actually contribute to these sexual antagonism, and what are their fitness effects? And what is the function of sexually antagonistic variants, and how do they actually generate these different sexually antagonistic phenotypes? Okay, so one way through which we can actually start to address these questions is by using a sex-limited selection approach. And the general principle for this is that the phenotypic and genetic change is normally quite prevented by the genetic constraints. So what we can do here is we can capture conflict by releasing constraint and allowing free adaptation in each of the two sexes. So, for example, in this case, you would have, you can have sex that is sex-limited selection in terms of we are not allowing or restricting selection in one of the sexes like male here, and so we are focusing on the female fitness side of the curve, so you're imposing or letting selection happen on the females. You can have this reversed as well, so you can restrict selection on the females, and then you can have it act on the males, right? So what we are doing in the group, and what I've been doing for the past couple of years, is using this kind of a sex-limited experimental approach. So we use a middle-class neighborhood design wherein we impose sex-limited selection. And we use our founding population that we use is quite the well outbred lab-adapted population, LHM, which a lot of people do. So what do we have in this instance is, first, I'll talk to you about the male limited selection. So here, the selection is limited to males. And how do we do that? Is we have 4 males. Well, it's 5 here. Yeah. It doesn't matter. You get the idea. And then there is only 1 female. So essentially, we are limiting selection to the males. Yeah? And the way we do it is we have this entire setup. So 4:1, or here 5:1, in one vial, and then we have them mate, right? Then we have the opposite thing as well, as in we have the female limited selection, where the selection is limited to the females, and here you can see there is more females, and there is just one male. So selection is free to act on the females, and we just have one male, so there's no selection acting there. So we're restricting selection in that sex. And again, this entire setup is in one vial, and we have 16 populations of each of these selection regimes. So we have each replicate population from each of these selection regime actually comprises of 200 individuals. And yeah, so in total we have 32 of these populations. Now, one really cool thing about this experimental design is that by having, as you can see, just one rarer sex in each of these, either the male or the female in each of these two sex-limited selection, we are restricting the selection to either one of the sexes, right? So that is really cool. The other really cool thing is we are accounting for the fact that each of these set up in one vial that you see contributes the exact same number of offspring every generation, right? And then we mix it up together, set them up again. So that's quite cool, and we have pretty good, well, I think we have pretty good replicative power because we have 16 replicate populations for each of these selection regimes. So it gives us a lot of power to see what's actually going on. Okay, and so what we've done is we have evolved them for 27 generations, and quite proud of that. Because in human year, it's well over, in human world, it's well over a year worth of work, which is pretty impressive. So that's great. So now that we have such a cool system, we can try and address the key questions that I was trying to talk to, that I showed you before. So what is causing these? Well, are there any sexually antagonistic variation? What are the fitness effects? What are the genomics, and what is the genetic basis of these sex-limited experimental evolution flies? Okay, so one of the things that I am going to show you is how we can characterize these sex-specific phenotypes. And what we do is, so fine, we've evolved these populations. Now we want to have a look at what are the phenotypic differences across the males and females in these evolved populations, right? So we do this by the competitive fitness assays. They are quite well characterized. So essentially it's quite cute in the sense we have our focal males or females from the 16 female limited and male limited populations, and with the founding LHM as our kind of control. And then we compete them with LHM browns. So the browns are essentially nothing but flies that have brown eyes that you can see here. They occur naturally. They're quite a recessive eye color, and we just maintain them using the same thing. So yeah, so this is the competitive fitness assay that we use. So to go in depth of how we actually do this is, so from each population, so remember we have 16 replicate populations where selection is only limited to females. We have 16 replicate populations, selection only limited to males. And then we want to characterize what is actually happening at the phenotypic level in terms of fitness in both males and females across all of these populations, right? So what we do, we call them the focals. There are focal males or females, so 5 focal males or females from each of the populations, compete them with 10 competitor LHM browns of either male or female, depending on which assay you're doing. And then we just have the other males or females of the LHM browns for them to mate with. Right, so this is the entire method that I use to do the competitive fitness assays to analyze or characterize the fitness effects. Okay, and all of this setup goes into a vial, and then we see the offspring counts. So essentially, I don't know what that is. Anyway, so essentially we look for the proportion of red versus brown. So if the red-eyed females or males are actually competing and doing better than the LHM browns, then you would see more offspring of that eye color. So that's why we use the brown and red, yeah? Okay, so I'm going to show you very preliminary results, and I say very because I just got it earlier this week, so we can all get a first glimpse together. So this is looking at male fitness. So here I am getting the focal males from each of the 32 populations and looking at what's actually happening to them. And you can see on the y-axis I have proportion of red-eyed offsprings, and on the x-axis I have the different replicates. So they are separated by the selection limited to females. And then you have the founder, and then you have selection limited to the males. What you can see here is there is a lot of variation, but also nothing is really happening. Like, we don't really see any significant differences in competitive male fitness across all these populations. Now, there is still another block running as we speak, so the data is not complete. So we will see what happens as I get more data out. In terms of female competitive fitness, though, what we do observe, again, same, the proportion of red-eyed offsprings, and then you have the replicate populations and across the two different selection regimes. What we do see is actually I have 60% of the data now. So based on that, I do see that the females from the selection limited to males population are actually doing a bit better. This might change when the 40% of the results come back in. Okay, so that's what happens here. And I'm just going to quickly move on to show you the other aspect of things that we are looking at. So we are looking at genomic signatures of of antagonistic selection as well. So here, as you can see, you have allele frequency on the y-axis, and you have multiple generations. So this is with my colleague Abhilash. He's a postdoc in the lab. And what we are trying to see is, and he does a lot of ABC simulations under the same experimental evolution setup that we have, and we are trying to look at the genomic signatures. So the green dots that you can see here are actually the time points at which the flies were frozen for pooled sequencing. So this is also happening as we speak. And yeah, so we're trying to see how the allele frequency trajectories change across these populations over time, over generations, and you would expect male beneficial alleles to rise in the populations where selection is limited to males, and you'll expect female beneficial alleles to rise in the population where selection is limited to females. And that's what you see in the red and blue over there. Well, with that, I'm going to thank my supervisors, Max and Ida, and Abhilash and Deborah, who are also on the ground with me. And thank you, everyone, for listening.
Okay, so I'm putting a timer, and at 40 minutes I will leave, so that we don't end up without coffee break. So hello, everyone. My name is Tiago. I'm a PhD student in parasitology in the University of Neuchâtel, Switzerland. And today I will talk about a specific microsporidian and how we can use it to control mosquito-borne disease. First, let's talk about mosquitoes a bit. So mosquitoes are more than just blood feeders. Actually, their primary source of food is nectar, like many other insects. They are pollinators, and only some species require the blood meal. And within these species, only the females will take a blood meal, and they need their blood to mature the eggs or produce higher egg clutch. And within this species, around 20% feed on humans. However, this 20% is a problem because of the disease they transmit. And during this talk, we will focus on two species of mosquitoes, Anopheles gambiae mosquitoes, that is the vector of malaria, pointer here. So this mosquito here, that is the vector of malaria. And we will also focus on one species of tiger mosquito, Aedes aegypti, that transmits other very important diseases. And because of the diseases, we have put in place many techniques to control their populations, mainly these three that are the most common. But the problem with these three techniques is that they use the same methods, insecticides, and in an evolutionary standpoint, insecticides is not a long-lasting technique because it leads to resistance to these insecticides. And because of that, we have been exploring other techniques, and 20 years ago, people were proposing microsporidians as agents of biological control. Microsporidians are obligated intracellular unicellular parasites that have been shown to infect several genera of mosquitoes, and that's why they were suitable for this vector control. And here I will focus mainly in Vavaria culicis, one type of microsporidian that we can see here in this picture. And actually, Vavaria will infect the mosquitoes during their aquatic stage, where larvae will just ingest these spores. And then the spores will infect the host cells, reproduce inside, leave the host cells, and infect new host cells. And the main mode of transmission of Vavaria is horizontally, either between larvae, when a larva dies and releases the spores, or if an adult ends up dying in the water, it could also release the spores in this aquatic environment. And previously, it has been proposed as a way of controlling these mosquito populations, first in Anopheles, because it was shown to stop the development of malaria. So when looking at the number of oocysts, they found that the number of oocysts was higher in the control mosquitoes, so mosquitoes that were only infected with malaria, than in mosquitoes that were infected with Vavaria and malaria. In addition to that, Vavaria culicis has also been proposed to control other species of mosquitoes because the way it affects the life history traits of these mosquitoes. And with that in mind, we have recently assessed Vavaria culicis infections dynamics in these two species of mosquitoes, so in Aedes aegypti and in Anopheles gambier. And the two key aspects of our design were that in our lab, we maintain Vavaria in both species, alternatingly infecting Aedes aegypti and Anopheles gambier to try to ensure it remains its generalist status. And also something that we did in this experiment and that we do in most of the experiments that we do in the lab is that we rear our individuals individually, which is a lot of work, but has also the advantages of giving the same infection dose to all the individuals and the individuals not interacting between them, which could also change the parasite load. And actually, we found that, first of all, to the best of our knowledge, we were the first ones to disentangle larva and pupa mortality that was previously reported as juvenile mortality. And that was interesting because actually when looking at larva mortality, we can see that individuals that were infected with Vavaria, so here in dark, they increased their larva mortality for each species, so here for Aedes and here for Anopheles. But when looking at pupa mortality, we realized that actually infection did not increase pupa mortality in neither of the species. And that was interesting because until now, and because of the life cycle of Vavaria, Vavaria was assumed to be a pupicidal parasite. And here with this data, we found that actually most of the mortality during the juvenile stage was larval mortality. In addition to that, we also found that Aedes aegypti, so here in the left, was able to clear the infection as adult. So when looking at the proportion of individuals that had spores, or that had enough spores to us to detect them, we can see that this proportion increases until day 10, where all the individuals had spores, and then decreases with time. And that was also interesting because until now, we thought that what happened is what we noticed in Anopheles gambier, where all the proportion of individuals increased until all the individuals had spores, and then it remained like that. So the results of this study highlighted that based purely in our results, Vavaria could be used to control disease like malaria, but its potential use cannot be extrapolated to disease such as dengue because the host seems to clear the infection. And also, it affects a lot the host, which could induce resistance towards the parasite. So with that in mind, we wanted to add a bit of an ecological aspect to this study. And in natural settings, host availability might be seasonal. So some authors had proposed that maybe the fact that microsporidians can infect different hosts is because they might use some hosts as reservoirs, so that if their, let's say, preferred host is not available, they can persist in another host and then infect later a new host. And with that in mind, we evolve our Vivraria. So starting with the Vivraria, our Vivraria colony, we evolve this parasite either exclusively in Aedes or in Anopheles, and we added another factor that was evolving or exposing the offspring of previously exposed individuals or previously unexposed individuals, and that's what we called exposed and naive hosts. And the results were that, first of all, when looking at larval mortality, we noticed that our lines of Vivraria didn't have any effect on larval mortality, or all the lines produce around the same larval mortality when looking at Anopheles gambier. However, when looking at Aedes aegypti, we noticed that the Vivraria lines that were evolved in Aedes had a lower mortality than the ones that were evolved in Anopheles gambier. And in addition to that, we also noticed that the larvae that the Vivraria lines that were evolved in a naive host induce a higher mortality than their respective treatments that evolved in an exposed host. So basically, the green bar plot, the dark green is higher than the light green, and same for the purple. In addition to that, we also realized or showed that no matter what Vivraria line we were looking at, Aedes aegypti still seem to clear the infection, as the proportion of individuals with spores increases a bit with time. In this study, we didn't find a time point where all the individuals had 100% of spores, but we also had less time points. And with all of that in mind, and if we put all the results together, both studies seems to highlight that Vivraria culicis could be used to slow or impede the transmission of malaria, but it doesn't seem to work with a disease such as dengue, as it affects too much the host, which could, again, produce or could lead to an evolution of resistance against the parasite. And also, the host seems to clear the infection, which is not a long-lasting. Yeah. And actually, I went super fast, so I think we will have time for questions. But with that, I would like to thank my lab, my supervisor, Jacob, the whole lab, the parasitology lab, the other lab with whom we share the floor, the biodiversity genomic lab, and actually my colleague Luis, who is presenting in this exact room in an hour. So if you want to hear more about Vivraria, virulence and transmission evolution, just come back in an hour. Thank you.
Very interesting talk, thank you. And I have a question about Wolbachia in your research. Did you take into account Wolbachia infection?
Wolbachia?
Yes.
I use Vivraria culicis. It's not Wolbachia. Maybe I misspoke. Yeah.
Antibiotic bacteria Wolbachia,
Yeah.
it can influence resistance to
Yeah.
some parasites in different species, and it's usually infected mosquitoes.
Yeah.
And did you take into account Wolbachia in your experiments on?
Normally, I mean, we use our colony mosquitoes so that we know they are not infected with Vivraria. So normally the Vivraria was with Wolbachia, sorry. So normally Wolbachia didn't play a role in the results. But I mean, I don't see how they could get infected by Wolbachia because they have been in the lab for too long.
If you
Yeah.
use in nature mosquitoes
Yeah.
are infected.
I mean, the study that we did was, so we didn't take it in consideration because it was with, yeah, it was not a factor that we took into consideration. Yeah.
For the context of
Yeah.
You have